will present from more maybe the user perspective, the use of AI, deep learning into the clinical practice. And the first one is Andreia Pinto of the Royal Brompton Hospital. Very nice to have you here, Andreia. Thank you for calling in. And she will talk on PCD8, how will artificial intelligence improve the diagnosis of primary ciliary dyskinesia. Andreia, the screen is yours.
Hello to all. And first of all, thank you for the invitation. I'm very happy to be here. I'll tell you about this tool. As Peter mentioned, PCD8 is a tool that we are using for our diagnostic. We are not using it still exclusively, but I'll guide you through it. So first of all, I want to do 2 disclaimers to this presentation. The first is that I'm still not sure how to call NOUS or NOUS, so I'll call it NOUS because I was told off once already saying it's not NOUS, it's NOUS. So I'll call it NOUS. I'm sorry for everyone else. And also I might refer to he instead of it because it's a very interactive platform and sometimes I just can't help it. So I'll start now by telling you a little bit more about primary ciliary dyskinesia. This is a rare disease, it's a genetic disease and a multisystemic disease. So primary ciliary dyskinesia is a disease that affects mainly the airway because cilia are present in all our body, but the place where it gives more symptoms is in the airway because cilia need to clear the airway from mucus and debris. This mucus serves to trap debris and bring them up or down in our throat. And if the cilia can't move them right, they will just be trapped and cause infection. So in this case, these patients have a very clinical, symptomatic board of symptoms. So that's why it is important to diagnose it carefully. So what we do for the sampling, we do a brushing, a nasal brushing. It's very similar to a nasal swab for a COVID test. So we try to target the inferior turbinate here in the nose with the brush. It can be a bit uncomfortable, but it's very easy to do. And then with this brush, we prepare it for several testing. One of the tests is electron microscopy, which is the one I'm going to be focused on, and we'll see why later on. The other one is light microscopy, where we see the cilia beating, and you will see, hopefully, in this wave, the cilia are beating and pushing the debris from the nose and from the airways to the nose. And then immunofluorescence, where we target for specific proteins that are involved in PCD, and genetics. So we need all these testings because PCD is very, very complex to diagnose. Electron microscopy is still a gold standard test to diagnose PCD because it's very specific and we can detect a lot from the images. So in here, for instance, you have cilia that are on top of the cells in a scanning electron microscopy imaging. But what we use actually is a transmission electron microscopy because we want to look inside the cilia. We want to look at the cross section. Like if cilia were hair, we would cross section them and look inside. And this is the beautiful cartwheel structure that the cilia have. And what we target, what we want to see inside is these doublets that look like 8 shapes. And the 8 shape in the center as well that we call the central pair from now on. Then the outer dining arms, which are these kind of hair protrusions like in these 8 shape doublets, and the inner dining arms, which are a bit more complex to see. Also, the radial spoke, which is kind of a net in the middle of the cilia to keep it all steady and straight, and the nexing links, which are very difficult to see, but when they are disrupted, the cilia get all disarranged. And I'll show you the defects that might be involved and interconnected with these defects in this next panel. So these are all the most common defects that are found in cilia. So here you have absence of outer dining arms. So those protrusion hair-like structures in the doublets do not exist. And this one you have absent of the inner dining arms. In this one, you have absent of both. Then you might have disorganization, as I told you, of all these components inside. Everything looks like a mess. And then you have more subtle defects, like just a translocation to the center of one of the doublets that mimics like if it was a central pair, but it's not, and the absence of the central pair. These are the most classic PCD defects. Those are the defects that actually classify a patient as having PCD. But there are also other defects that are considered secondary defects. And those are, for instance, the presence of multiple cilia inside the same membrane, or the presence of single tubules, like lost in the middle of the cilia, and a broken central pair where only one tubule is visible. These defects also contribute for the analysis, but they have to be considered as secondary. They don't contribute for the disease. And why is this important? Because all of us, when our cilia are being in some sort of aggression, for instance, if you have a cold, if you are in the middle of the Antarctica and very cold temperatures, if you have hay fever, this can happen to your cilia. They can become a bit abnormal. What happens is they just ciliate and form normal cilia, and new cilia and perfect normal cilia. But in the PCD patients, the ones that have genetic problems, these will happen all the time, even if the cells deciliate and become nude and reciliate again, the cilia will always be defected. And this is why it's so important to have this library of defects and knowing how they look. So with the help of Cosmonio, now held by Intel, now part of Intel, we were equipped with this machine learning tool, which was the NOUS. And we have actually in our hospital a learner interactive AI workstation where we are feeding all these images of patient data containing hundreds of cilia. And we were teaching the machine what is a normal cilia, what is abnormal cilia, and cilia that are useless, cilia that we cannot actually use for a diagnosis, because that also can happen, and that contributes for our final decision. So in this small clip here, and sorry for the quality of the clip, but I really wanted to see my hand doing this, using the machine. So I do draw a box around a cilia that I'm interested in detecting, and then I detect it as a cilia using the platform. It's really hands-on approach, user-friendly, and has an instant feedback. So if we do this, soon we start having suggestions from the software saying, okay, so this might be a cilia as well, because taking into account what we are feeding the system. So after this, we take these detected bounding boxes into a classification project. And in the classification project, you will only see these crops of cilia, and they will be classified. So we can train them. We train them in which label they are, if they are abnormal, normal, or useless. And then the machine will start giving suggestions. So if this clip in the upper left corner here, you will see some stickers, pink, blue, and yellow. Yellow is useless cilia, pink is abnormal cilia, and blue is normal cilia. And what we see here is our stream of training, basically. This is what we did to train our system using all these images. So just to give you a bit more of how the platform works and how does it look, this is an overview of an image of a patient in one of our projects. We have in yellow all the cilia detected, so with the yellow sticker, and then the blue and the pink stickers are the classification into normal and abnormal. And then we have the yellow stickers, which for us are a bit less interesting for posterior analysis, which are the useless. And this is how we will see. So in the right hand here, we see the media. So all our images, and if they are already trained or not. If they have a green sticker, it means they are trained. If they have a yellow sticker, it means they need to be trained or accepted the suggestion done by the software. So just an example of how this works. So you all know probably by now that this works in the black box approach. So we really don't know exactly how this training is happening inside, but we can actually sneak peek inside. And for that, we use a tool called the Saliency Map, which is here. I just click and appears in the yellow arrow down here that I can't see because of, okay. And when I click in the saliency map, it will give me the where the system is paying attention to. So in this case, just going back again, we detected this cilia. The system was 87% sure that this was a cilia. I agree. And it was 92% sure that this cilia is abnormal. So just to remind you that the normal cilia have to look exactly like this. So at first sight, this looks like a very normal cilia, but when we turn on the attention map, we see that it's giving attention to this bit in the center here. And actually, this bit in the center is a protrusion of the central pair, which is classified as a very subtle defect, which is the C2b absent. And when the C2b absent, and I have like a scheme here, and sorry for being so I'm being a bit detailed on this genetic bit, but I really wanted to tell you that when this protrusion here of the central pair is missing, we might have like a hollow bit on the cilia here. And this was actually detected by the machine. If you look at this side, it looks a bit more filled, a bit more dense, and this side is a bit more hollow. And this was detected, and this is really good. It means we're going forward when classifying more subtle defects, not only defects that are more clear or more frequent. Okay, so I will go a bit more about this later on. I just wanted to tell you how are we setting up a project right now. So we create a task chain. We start by creating a detection project where we detect cilia. Then we export those detected bounding boxes, those cilia, into a classification project where we classify them in normal, abnormal, and useless. And inside the abnormal, we will have a second classification. And here we can have as many labels as we want to classify it with what is actually the defect. So why is it abnormal? So what is the defect? What does it have that makes it abnormal? In this case, you have like a stream of projects. In this test one project, we have cilia detection, normal, abnormal, and useless. And then we have two extra labels here, which are outer-dining arm defect, which is that the small hair-like protuberance in the doublet, and another other defect that can be anything else.
Andreia?
Yes.
There is a question in the chat by Sunil, who's asking what is the useless sticker?
Okay.
What is the meaning of that? Is it also a cilia image, and why is it useless?
Okay, very, very good question. I will mention it later on, but I can tell you exactly. Well, the reason I didn't went into much detail is because it's the least interesting, but it's actually very important. So a useless cilia, for instance, if you see this screen here, this 3 cilia here, they are useless because we cannot see any detail. If we cannot see the detail, we cannot analyze that cilia. So it might be tilted, it can be out of focus, it can be just incomplete, it can be like 2 cilia in the same bounding boxes. Those are all useless cilia. And this is something that we as human, we don't do. We don't classify into useless when we are doing an analysis, but the machine needs it. And we discovered this a bit later on. It was really, really interesting because this is the learning curve, right? So we do need to create this label. This label is really important. It's as important as normal and abnormal. So when we have these cilia here, they can still be detected, but they will be just put in a useless label. I hope that it clarified it for now. We can talk a bit about it later on more. Okay, so I want to show you now how we do a training for a child label. The child label is these second classification labels that we have. So for us, the only second classification that matters to us is the abnormal label, because there's where we have the abnormalities. A normal cilia is a normal cilia. It has 9 of those H-shaped structures and the central H-shaped structure. An abnormal cilia can have a lot of classifications. That's why it's the only thing we subclassify. So for instance, in this video, we have this cilia here. I am detecting it as a cilia, and then classifying it as an abnormal cilia, and then as an outer dining arm and an other defect, because this cilia is very disarranged. It's all messed up. So this has a multiple subclassifications. So on top of the fact that I can subclassify my cilia, the abnormal class, I can also give it multiple labels. I don't need to give it only one label. Because to be honest, there are cilia that can have four second classifications, four child labels, and this is really, really good, because this is the way we do it when we are doing the diagnosis ourselves. We just tick boxes, and here we just classify it, multi-classify it in multiple labels. So now going through a more realistic approach. So this is actually what we are doing now. So this is our reality at the moment. So we have our three main labels: useless, normal, and abnormal. And inside the abnormal, we have around 14 labels. We can't see them all here, but we have like around 10, I think it's 10 child labels at the moment. We're always playing around and see what kind of results we can have. But this is what we are working now for now. And we have outer dining arm defects, inner dining arm defects, disarranged, several types of disarrangement, because if you remember, I referred to the fact that we have secondary defects that might look like disarrangements, but they are very specific type of disarrangements associated with secondary defects and not with the disease itself. This comes with experience, so you know how to classify them in each group. And that's why I am doing myself, because I am used to see these defects, and I can train the machine with my own knowledge. So these are the labels we are using right now. And just to show you how is it behaving, and this is already not training but analyzing. Well, it is training because I'm accepting the suggestion, but you can see that the NOUS is already analyzing and giving me a feedback, and this is what we see here. So in this case, I will select the cilia. This cilia is abnormal and have outer dining arm defects, and the NOUS is 100% sure of that, and I agree, it's true. And this one the same. Okay, and this one is useless, and it is also correct. So this is how it goes now. So it gives suggestions, and I just accept or reject. And I'll tell you about the performance in a bit later on. Okay, so we could stop this presentation right now because it's working really well. We can spread it to the world, share it with everyone that does PCD diagnosis, but we still have something to cover and something to worry about, which is subjectivity. Okay, so this cilia, for instance, obviously we're not in the PCD meeting, so I cannot ask for your agreement, but I can tell you that if I show this cilia to 6 experts, they will all probably agree with me, or 99% will agree with me, that this is a normal cilia. It has 9 doublets, that's how we call them, and a central pair. And it's very good cross sections, it's very detailed, we can see everything. But on the other hand, this cilium is tilted. But for me, when I analyze it, I can still see some of the features. I see a central pair, I see some of these outer dining arms. I also can see some of the inner dining arms. So I would say it's tilted and it's a very dodgy cilia, but it's still analyzable. I could still use it. But maybe some of my colleagues would disagree. And this is not uncommon. This might happen, and this happens, okay? So I started to think, okay, but I am training this machine, so maybe I'm biasing the machine with my own knowledge, okay? And I really wanted to put this here to sensitize for the subjectiveness and perception of the human mind and how this might be impacting this training and, in the end, all the results. Okay, so quickly, if I just use the chihuahua or muffin example that you probably all already seen, is a very good example for machine learning and training. So as a human, when I look, I might be confused, but after a couple of seconds, I know what are a muffin and what is a chihuahua. But for a machine, I would have to give a lot of chihuahuas and a lot of muffins for the machine to start to understand what is a chihuahua and what is a muffin. So this is exactly what we do. We give a lot of cilia of all the classification labels. We cannot only give one, otherwise it won't be enough and the machine won't be accurate and don't perform well. So on top of this, I was also thinking, as Anna mentioned, this is my own feeding. So I am feeding the system with the chihuahuas and with the muffins, and I am telling the system exactly what is what. Obviously, cilia, normal and abnormal and useless. So to be sure that I was doing this right and we are not creating a beast, I'm sorry to refer to it like this, but it was. So I challenged my colleagues, all the experts I could gather, to look at images of cilia that NOUS already analyzed. And what I wanted with this is to see if there was agreement between all of us, of what sort of agreement there is between all of us, between me and the NOUS, between the NOUS and the experts, and between the experts and me, and between the experts all between themselves. So I gave these experts 600 images of cilia, all from these 3 labels. So here we have exactly what was asked to the experts. So to classify as abnormal if they have any of these abnormalities, to classify as normal if they were completely normal, no doubt about it, and as useless if they were just unreadable, if they were not suitable for diagnosis. And in the end, luckily and very fortunately, we did have a very similar, I'll give you all the information now, and I'm going through it quickly so it doesn't get too thick. So in the end, we all agreed with each other around 80%. So me and the NOUS and the experts between themselves. And even in the different categories, obviously the useless is the one that had a higher score, but it was very good to see that the abnormal category reunited an agreement of 80% between everyone. And even in very difficult cases, for instance, in this graph here in the middle, the first column is for inner dining arm defects. Inner dining arm defects are the most challenging ones, are the ones, the inner protrusions of these H-shaped structures. And these are always always the defect that gives a lot of disagreement and arguing between the experts. And in this case, the experts didn't agree as much as with each other, but the NOUS gave, so the experts didn't give 100% credibility that there was an inner dining arm defect, but the NOUS gave. So the NOUS was very accurate in that analysis. And this was very, very, very good. So we're very happy with this results, and we want to take it further and do it again for all our other projects with the multi-labels. So now we want to challenge our experts with not only classifying it into abnormal, normal, and useless, but also inside the abnormal in all the categories. And this is something that it's being done. At the same time, we are using our system alongside the diagnostic meetings. So the diagnostic meetings are meetings that occur every 2 weeks, where a multidisciplinary team discuss all the tests, the videos of cilia beating, the immunofluorescence, targeting proteins, the genetics, and the electron microscopy. And in these meetings, there is a new player. So NOUS is playing as well, is giving it or his results for each patient. And until now, we have like around 99% of success with matching what the expert think when he's analyzing this data in the service, in the clinical stream. So this is really, really good. And this is what we are doing. And this is kind of a validation process, which is giving brilliant results. So just to wrap up and to tell you a bit more of what we want to do next and what we want to do with this tool. So we obviously want to reduce this diagnostic turnaround time, and I'll tell you how. So a specialist can take 45 to 90 minutes to screen a patient. It depends on the complexity of the case. And then it needs extra time to do a report. For the NOUS, if we have an automatic acquisition of all the cilia and images, like a high throughput analysis, uploading a patient to PCD takes less than a minute. So it's really, really promising. So we really want to improve the detection of subtle defects. As I showed you with the attention map, we are already going to it, but obviously this is one image and sometimes we don't have the expected result yet, but this only requires more chihuahuas and more muffins. So this is what we are doing now. We are making it even more specific and detecting all these subtle defects that we want to be able to detect. Obviously, I have to be able to detect them myself. It I can't detect them myself, I won't be able to feed it to the machine. So this is the realistic part of it. So the central pair protrusions is one, and the inner dining arm defects are another one that we already have very, very good results. We want to roll it out to the other UK centres. Currently, there is Leicester, Southampton, and London where we are, and we are talking to them about this. They know where we are, and they're very curious about it. And we hope they can be using this model soon as well. And just the last thing I want to mention is that we want to go further. So if the electron microscopy analysis is considered subjective because we have so many gray areas that we are working on, we have an even more demanding area, which is the videos of the cilia beating. And this have a range of in the specificity that can be huge. And a human eye obviously can only be focused and concentrated for a short amount of time. So we cannot be acquiring images, videos of cilia beating for a whole afternoon. You have to rest, you have to be focused and completely clear your mind. So with NOUS, we hope the next stage will be to be able to do this, to be able to analyze the cilia, the flow of the mucociliary clearance, this clearance of mucus by the cilia. So we hope we can do this using these videos, and we are working on it, and hopefully we'll start on it very, very shortly. So this is it. I want to thank everyone that was involved in this. I was a bit over time. I'm so sorry. I just realized. I want to thank everyone that is in this slide. PCD from the service from the Brompton, Cosmonio, Intel, and Diatom for providing some of the equipment that we are using, and the NIHR obviously for funding. And feel free to contact me and to make any question you want. I will stop sharing, otherwise I can't see anything.
Thank you very much, Andreia. You indeed went a little bit over time. We already had a question in between. Yeah, you were. It is a very interesting to see the real application of something like this. So I thought it was good to allow you to finish the wrap up and to show the future that you see and so I should not do any questions, but move to the final presentation of this afternoon. Thank you very much. There are some questions in the chat, so maybe you can
I can answer them.
Give your answers later on, or maybe people can contact you later on. So thanks again, and we move out here and go to the next
I'll answer
lecture.
them. People find me.
Okay, thank you.
Thank you.